today we will be talking about the identification of fossils and the codes of systematic nomenclature. Fossils in any rock, it is characterized by some features. You identify any particular fossil based on some peculiar features. These features enable the fossil to be placed in a taxonomical framework, and that is what is meant by its identification. That means, the identification of fossils, or to identify a fossils, we mean that the fossil should be placed in a suitable or a particular taxonomic framework. There may be a variation of the precision to which the fossils are identified. That means, different fossils are identified based on different levels of precision. However, species designation is usually unnecessary. It may, for example, be possible to establish a marine origin for the sediments simply by noting the presence of fossil remains of cephalopoda. That means, wherever in whichever rock you find the fossils for the cephalopods, that is enough and sufficient to assign a marine origin to that rock. Or you can get the fossils of echinodermata. So, if you can identify the fossil as an echinoderm, then also you can assign a marine origin. That means, in that case you do not need to go to the species level. Paleoecologic analysis given in a well understood stratigraphic framework, it seldom requires the species identification, as I told you. Similarly, in stratigraphy, it may be possible to distinguish between identical rock units of differing age by fossil identification. That means, two same rocks, that means two shales, they may look similar. For example, you can have two black shales, but you may have different fossils into those rocks. So, lithologically the rock is same, but biologically or paleontologically it contains different fossils. So, it belongs to different ages. So, you may identify the fossil as a phylum, a class, or an order. In establishing a classification, lower taxonomic categories are generally delineated first, and then they are assigned to the higher categories. The procedure is normally reversed in the fossils. Like here, you get a fossil. For example, if you get a fossil covered with polygonal plate like elements, they are all arranged radially. You have the columns of the plates, they extend from one pole of the roughly spherical skeleton to the other. Most plates have one or more knob-like structures called as tubercles, which by analogy to the living relatives are points of attachment for movable spines. This combination of the characters suggests that the specimen belongs to the phylum Echinodermata and within it in the class Echinoidea. No other class of this phylum bears such characters. We thus eliminate at sight all but one class, thereby reducing the identification problem to choice among thousands of species. Phylum identification is therefore the initial step and is important to give the full working knowledge of the taxonomic characters most useful in distinguishing phyla. There are approximately 12 main phyla in the fossil record, and each is identified or defined by an average 6 to 10 characters. Coming to module 1, which discusses the evidence for speciation and the patterns of species multiplication and associated morphological change as revealed in the fossil record. Speciation is regarded as the key stage in the generation of the biological diversity. That means, how would you produce the biological diversity in an ecosystem? By the initiation or the production or the evolution of new species, because it is the point at which the reproductive isolation is achieved. The hallmark of the biological species is the speciation only. Many studies of the modern species concern themselves with the short-term genetic fluctuations between populations of a single species. How those fluctuations may come about and how they are maintained. Such studies are not possible in the fossil record. On the other hand, the fossil record documents extended time scales and may therefore identify patterns of long-term changes relevant to different theories of speciation that are unavailable in the recent world. Irrespective of the mode of speciation, the fossil record demonstrates repeated instances, geologically speaking, of sudden increase in the number of species, and these are usually associated with rapid morphological divergence. Such events are commonly referred to as the radiations, more usually adaptive radiations, and are inferred to have come about by causes. What causes? Intrinsic to the organism and or extrinsic causes. Although studies of the radiations involving populations and closely related species are studied in the recent world, broad scale pattern can only be studied through the time dimension supplied only by the fossil record. Now, let us discuss the species recognition and speciation in the fossil record. There is a plethora of species definitions and criteria for recognizing species in the recent world, but few are applicable to the fossil record, where only a limited amount of morphological data, usually the skeleton, is available for study. That means, you have only the skeleton or the hard part available in the fossil record to study. The biological concept of Ernst Mayr is probably that most favored by people studying the recent world. Species status is achieved when populations diverge genetically to such an extent that the reproductive cohesion is broken. There may be several reasons for the fragmentation of the reproductive continuity. It may be geography, that means if two species or individuals of the same population or two populations of the same species, they start to live in a geographically different area, in a different ecology, and if they start behaving differently, very naturally the reproductive isolation will come at some point or the other. Although Mayr stressed geographical isolation, the parental species may continue to live alongside the daughter species. In practice, most modern species are not recognized on reproductive criteria. Instead, some measure of morphological, genetic, behavioral or ecological difference is used as a surrogate for reproductive incompatibility. As such, paleontological species recognized almost exclusively on morphological differences and are equally as valid as modern species. In fact, there has been empirical justification for equating genetic differences with morphological differences between modern species of some group such as bryozoa and Darwinian finches. It is, however, recognized that morphological differences may not always indicate species differences. For example, sibling species or polymorphic species including mimetic species. The evolutionary concept of George Gaylord Simpson, they define a species as an ancestor-descendant sequence of populations changing through time with their own trends and tendencies. This definition embraces asexual species. Such a definition allows for one species to change gradually to another. This is called as anagenesis, as well as instances where a species is budded off or where one species splits into two or more, which is called as cladogenesis. The Henningian species concept, which is named after the German entomologist Willi Henning, like the biological species concept, is based on the criteria of reproductive isolation. However, the species limits are recognized only at the point where one species splits into two or more and the interbreeding pattern of gene flow is disrupted, at which point the ancestral species cannot by definition live alongside the descendants. The species definition is strictly dependent upon the shape of the phylogenetic tree. Now, let us discuss the recognition of species. Species in the fossil record are nearly always recognized on the morphological criteria, as you may be knowing. There are some exceptions, such as instances where species are distinguished from one another purely on the basis of the stratigraphic occurrence of or geographic location, but these are usually accepted as stopgap measures, to be revised when more information about the morphology becomes available. Monophyletic species are recognized on the basis of possessing one or more unique morphological characters, and this corresponds most closely with the Henningian species concept. Species viewed in this light have the same ontological status as higher taxa such as genera, families, orders, etc. In reality, very few species can be recognized in this way, and logically it denies the existence of ancestors, since ancestors have no unique features. Typological species were once commonly recognized, whereby a single specimen, usually the first discovered or the most complete, was chosen as the Linnaean type, and this formed the nucleus of morphological variation considered worthy of species status. How much variation was to be allowed before a new species was recognized? It was never stated. Such species are usually frowned upon now as being steeped in the philosophy of pre-Darwinian essentialism. Yet, there remain many instances where such species are still used. Phenetic species arose out of numerical taxonomy. Here, species are measured for as many morphological continuous variations as possible. The variables are then subjected to multivariate analysis, and the clusters or the species are then recognized. Although this may appear to be objective and the theory free, and it is heavily dependent upon the original samples analyzed and the multivariate analysis algorithms used. It is not always easy to identify which of the individual variables is contributing to the differences, and therefore, it is difficult to diagnose the species. Phylogenetic species are the smallest diagnosable assemblages of specimens which show a unique combination of characters. Combination is the operative word here, since there is no requirement for identifying unique characters. For example, monophyletic species recognition. Of all the recognition criteria, this most faithfully agrees with all the process-based definitions of species, as well as agreeing with the day-to-day practice of paleontologists. Estimation of species longevity may depend on which of the above ways of the species have been recognized. Surveys across various fossil groups give average figures that range from millions of years for small mammals, where generation time is very short, to 13 million years for the dinoflagellates, which are largely asexual. Speciation in the fossil record is generally accepted that most speciation events, with the possible exception of those caused by hybridization and polyploidy or chromosomal duplication, are top or too protracted to be detected in the recent world and too brief to be seen in the fossil record. The lower limits of the time that can be successively sampled in the fossil record appear to be something like 5,000 to 10,000 years, and such deposits are very rare, restricted only to the lacustrine or the lake deposits, which is of relatively shorter duration and small geographic range, and some deep sea deposits also. Nevertheless, there have been many studies seeking to establish the precise pattern of speciation. There are two polarized theories on the tempo and pattern of speciation, punctuated equilibria and phyletic gradualism. Both have support from theoretical and empirical studies in the modern world, and it is likely that both modes of speciation can happen. Phyletic gradualism, something which you can understand that it holds that morphological evolution is gradual and it occurs independently of the speciation. Most of the species or studies they support that this mode of evolution involved organisms that can be minutely sampled through the continuous rock sequences. Some gastropod species they show an example of changing or replacing another in the ancestor descendant sequence, that is the gradual speciation by anagenesis or by a shift in the modal shell form as measured by the morphometric discriminant analysis. In this example, the gradual change has been correlated with the increasing depth of water. The populations intermediate between the two species were presumed to have lasted for about 73,000 to 2,50,000 years. In this pattern, there is no rapid shift in the morphology, and in studies of this kind, the species limits are usually recognized by comparing the morphological variation over period of time with morphological variation of modern species and dividing up the continuum accordingly. In another study involving radiolarians recovered from Pacific Ocean deep sea drilling cores, an instance of speciation was detected occurring during the periods of about 500,000 years, and in this case both the ancestral and daughter species showed marked deviation in many aspects of skeleton form. Further, it is interesting to note that morphological deviation, although slightly accelerated during the speciation event, continued for some time after the separation of the morphotypes. What it suggests? It suggests strongly that morphological evolution is decoupled from speciation. In this instance, both parental and daughter species showed morphological evolution, and it may have been possible to suggest that single ancestral species gave rise to two daughter species. The author of this study refrained from this because the ancestral species which still lives today shows a high degree of polymorphism and geographic variation, the scale of which is comparable to the variation seen in the lineage. In this study, it needs to be pointed out that many but not all of the studies that support phyletic gradualism report information on continuously varying morphologies such as lengths and shapes. These parameters are inherently gradualist in their measurement. Unlikely many characters are used cladistic analysis such as presence or absence of 4 toes versus 5 toes, in which there can be no intermediates. Then comes the taxonomy and the concept of nomenclature. Taxonomy is a fundamental and dynamic science, as you would have seen. It is dedicated to exploring the causes of relationships and similarities among organisms. Classifications are theories about the basis of natural order to identify a fossil form correctly, and indeed are keys to find out further information about it. Sound classification and nomenclature lies at the foot of all biological and paleontological work, without which no coherent and ordered system of data storage and retrieval is possible. Taxonomy or systematics, as it is sometimes known, is the science of classification of organisms and is the oldest of all the biological disciplines. It might surprise you sometimes also. In the formal nomenclature of any species, living or fossil, taxonomists follow the biological system of Linnaeus, whereby each species is defined by two names, one generic and the other specific. For instance, all the cats, large or small, they are related, and one particular group has been placed on the genus Felis. Like for example, the cat is also Felis, the lion is also Felis. Of the various species of the Felis, the specific name Felis catus refers to the cat, and Felis leo refers to the lion, and Felis pardus it refers to the leopard. In the full taxonomic nomenclature, the author's name and the date of publication are given after the species. For example, Felis catus, to which you can put Linnaeus 1778 in the bracket. In paleontology, it can never be known for sure whether population with a particular morphology was reproductively isolated or not. Hence, the definition of the species in paleontology, as in most living specimens, must be based almost entirely on the morphological criteria. Moreover, only the hard part of the fossil animal are preserved, as much useful data is vanished. A careful examination and documentation of all the anatomical features of the fossil has to be the main guide in establishing that one species is different than the related species. In very rare cases, this can be supplemented by a comparison of the chemistry of the shell also, as has proven especially useful in the erection of higher taxonomic categories. Within any interbreeding population, there is usually quite a spread of the morphological variation. On a broader scale, there may be both geographical and stratigraphical variations, and all these must be carefully documented if the species is to be ideally established. Such studies may be very useful in evolutionary paleontology. When a paleontologist is attempting to distinguish a species in a newly discovered fauna, say of fossil brachiopods, she or he has to separate the individual fossils out into the groups of morphologically similar individuals. There may be 8 such groups, each distinguished by a particular set of characters. Some of these groups may be clearly distinguished or distinct from one another. In others, the distinction may be considerably less, increasing the risk of the greater subjectivity. These groups are provisionally considered as a species which must then be identified. This is done by consulting paleontological monographs or papers containing detailed technical descriptions and illustrations of previously described brachiopod fauna of similar age and comparing these species point by point. Some of these species may prove to be identical with already described species, or show only minor variations of a kind that would be expected in a local variant within the same species. Other species in the fauna may be new, and if so, a full technical description with illustrations must be prepared for each new species, which should be published in a paleontological journal or monograph. The description is based upon the type specimen, which are always thereafter kept in the museum. Usually one of these, the holotype, is selected as reference specimen, and the fully illustrated comparatively details may be added from the other specimens, and this is what you call as paratypes. There are various other kinds of specimens also. For example, the new type. New type may be erected when the holotype has been lost, or when the species is being re-described in a fuller and more up-to-date terms when no type specimen has previously been designated. A new genus will be designated as gen nov by its author, and following the generic name, a new species sp nov and new subspecies ssp nov. The new species must be named and allocated to an existing genus, or if there is no described genus to which it pertains, then new genus must also be erected. Coming to the taxonomical hierarchy, although all the taxonomic categories above the level are to some extent artificial and subjective. Ideally, they should as far as possible reflect the evolutionary relationship also. Similar species are grouped in genera, singular genus, or genera into families, families into orders, orders into classes, and classes into the largest division of the animal kingdom, that is phylum or phyla. Phyla is plural and phylum is singular. There may be various subdivisions of these categories, for example, superfamilies and suborders, etc. And in certain groups, there is even a case of erecting superphyla. There are only about 30 phyla in the animal kingdom, and only about a dozen of these, for example, mollusca and brachiopoda, leave any fossil remains. The basic principle is that morphological and other similarities reflect phylogenetic affinity, which is called as homology. There is always so, unless for other reasons similarly or similarity results from the convergent evolution. But in assessing similarities, how does one decide that which characters are important? How should they be chosen to minimize subjectivity and produce natural order groups? There is no universally accepted method of facilitating these, and the taxonomist has used different methods. In recent years, three sharply contrasting schools have emerged. These are the schools of evolutionary taxonomy number 1, numerical taxonomy number 2, and cladism number 3. In erecting a hierarchical classification, taxonomists used a traditional and very flexible combination of criteria. First, there is morphological or phenetic resemblance, the extent to which the organisms resemble one another. Second, phylogenetic relationships are, along with the phenetic resemblance, considered important. By this is meant the way, as far as can be determined, in which the organisms are actually related to each other, that is in terms of recency of the common origin, which of course grades in the evolutionary taxonomy. The order of succession in the rock record and the geographical distribution may play an important part in deciding relationships. This practical approach to taxonomy, which took all the factors into consideration, has for a long time been the backbone of the paleontological classification, and is still being considered to be the best method by stratigraphic paleontologists or stratophenetic paleontologists, who place much emphasis on time in seeking ancestor-descendant relationships. For some taxonomists, however, the uncertainties and subjectivity which are almost inescapable in any kind of the classification seem to be particularly acute in classical taxonomy, as did the limitations of the fossil record in terms of preservation. Numerical taxonomy has been found very useful in some instances, but subjectivity cannot be eliminated, since the operator has to choose how best to analyze the measurements made and may need to weight them, giving a new character or a few characters more importance in order to obtain meaningful results. Hence, the objectivity of the numerical taxonomy is less than it might appear. The third school relies upon the phylogenetic criteria alone, emphasizing that features shared by organisms manifest in nature a hierarchical pattern evident in the distribution of characters shared amongst the organisms. It is known as cladist or phylogenetic systematics. Thus, in closely related groups, we would see shared derived characters or synapomorphies, which would distinguish this group from others. The central concept of Henning 1966 was that in any group, characters are either primitive, that is symplesiomorphic, or derived. Thus, all vertebrates have backbones, and the possession of a backbone is the primitive character for all the vertebrates, and is of course a derived character as compared to the invertebrates. Synapomorphy and the symplesiomorphy thus delineate the relative status of a particular characters, with respect to particular problem. Henning endeavored to provide an objective methodology for determining recency of common origin in related taxa based on the primitive and derived characters. Such relationships are expressed in a cladogram, in which the dichotomous branching points are arranged in a nested hierarchies. Suppose a taxa A and B share a unique common ancestor. They are said to be the sister groups. They both share an evolutionary novelty or synapomorphy, not possessed by a taxon C. Now, C is the sister group of the combined taxa A, B and D, and D is the sister group of the combined taxa A, B and C. In performing a cladistic analysis, therefore, a taxonomist assumes that dichotomous splitting has occurred in each lineage and compiles an unweighted character data matrix. The more characters and character states that there are available, larger the database, and larger databases are often routinely processed. How can we distinguish symplasiomorphic, that is shared primitive character states? The most useful way is an out-group comparison. Here, an in-group of which the relationships are under investigation is specifically designated and compared with a closely related out-group. Any character present in a variable state in the in-group must be plasiomorphic if it is found in the out-group. Likewise, apomorphic characters are only present in the in-group. Henning distinguished three different kinds of the cladistic groupings. Monophyletic groups contain the common ancestors and all of its descendants. Paraphyletic groups are descendant from a common ancestor, usually now extinct and now known as the stem group, but they do not include all the descendants. Polyphyletic groups, on the other hand, are the results of the convergent evolution. The representatives are descendant from different ancestors, and hence, although these may look superficially similar, any polyphyletic group comprising them is artificial. A cladogram is not an evolutionary tree, it is an analysis of the relationships. As such, it is a valuable and rigorous way of working out and showing graphically how organisms are related, and it forces taxonomists to be explicit about the patterns and groups. The methodology of the cladistic is especially good when dealing with the discrete groups with the large morphological and stratigraphical gaps, and to these it brings the potential for real objectivity. So, where does that leave the potential contribution of stratigraphy in reconstructing phylogeny? Whilst a few transformed cladists negate the value of the fossil altogether, successive appearance of taxa in stratigraphy cannot be denied as an essential source of data. However, imperfect the fossil record actually is, very commonly there is an excellent correspondence between the cladogram and the rock record. On the other hand, the combined cladistics and biostratigraphic approach may throw up unexpected patterns. The ultimate problem, not only for the cladism, but for all taxonomic methodology, it results from the convergent evolution. Resemblances in characters or character states may have nothing to do with recency of the common origin, but from convergence it may not always be possible to disentangle the results of the two. Even so, cladistic methodology coupled with the biostratigraphy seems to be the best way forward, an essential prerequisite for drawing up the meaningful phylogenetic tree. I hope I could make you understand how to identify the fossil and the basic concepts of the systematics. Other things will be discussed in the other lectures to come. Till then, thank you very much.